let us understand the basic pathophysiology of hypertension. Now, to understand the physiology of hypertension, we need to understand the four different parameters which is affecting the human blood pressure. So, the first and important is humoral mechanism, which is comprised of renin-angiotensin-aldosterone system. Second is neuronal regulation. Third is peripheral autoregulation, and fourth is electrolyte balance. So these are the four parameters which are responsible for maintenance of normal blood pressure. So when the condition are such where the blood pressure goes down, these four system try to restore the blood pressure. When the condition where the blood pressure goes up, these four system try to decrease the blood pressure and normalize the blood pressure. So in a nutshell, I can say that these four mechanisms are the homeostatic mechanism to maintain the blood pressure throughout the human body. Now, when there is a disturbance or malfunction in this four system, it leads to persistent elevation in blood pressure more than 140 by 90 mmHg as per the JNC 8 guideline. So I have narrated the four different mechanisms which is responsible for continuously elevated elevation in the blood pressure. The first is malfunction in humoral mechanism. As I said, humoral mechanism is mainly focused in the renin-angiotensin-aldosterone system. In short, we can say RAAS system. Second is abnormal neuronal mechanism, where sympathetic and parasympathetic pre and post ganglionic neuronal secretions affect the blood pressure. Third is defect in peripheral autoregulatory mechanism, where peripheral autonomic nervous system, which is a major component of peripheral nervous system, it is having some abnormality, defect. And the third is disturbance in the serum electrolyte concentration, intra as well as extracellular. So these are the four pioneer mechanisms behind the rise in blood pressure, which we will discuss one by one in detail. To understand the pathophysiology of hypertension, as we discuss, we need to understand the humoral mechanism first. Malfunction in the humoral mechanism is the major cause of persistently elevated blood pressure in the patient suffering from hypertension. So let us understand the renin-angiotensin-aldosterone system first. Now, what is the function of renin-angiotensin-aldosterone system? As we discussed previously, renin-angiotensin-aldosterone system is the normal regulatory homeostatic system in the human body to control the blood pressure in the different situation when the blood pressure get fluctuated. If the blood pressure is high, this RAAS system stop working and blood pressure goes down. When the blood pressure goes down, the RAAS system gets activated and the blood pressure will rise. I will give you simple example. In the condition when the patient is bleeding, when the patient met with an accident and patient is bleeding and losing the blood volume gets down, and during that condition the perfusion in the vital organs gets down because of the low blood volume and low blood pressure. During that situation, the RAAS is the life-saving system which gets activated, increase the perfusion in the vital organs by increasing the blood pressure. So this is how the RAAS system is very helpful. But as I said earlier in the pathophysiology, malfunction in the humoral mechanism, where in spite of having normal perfusion, the blood pressure keeps on increasing due to some local as well as systemic malfunction, and those malfunction we are going to discuss in this lecture. So initially, first of all, we understand renin-angiotensin-aldosterone system. We need to understand the renin. Now, what is renin? So the renin is nothing but one type of enzyme. This enzyme is basically secreted in four different conditions. So let us understand the four different conditions where the renin start secreting. And once the renin starts secreting, which is an enzyme, it is increasing the blood pressure by activating the RAAS system, that is renin-angiotensin-aldosterone system. First situation. Now, if you understand, this is a normal circulation of a human body. We had discussed the normal circulation in the previous video. If you have not seen that video, go back to that video and understand the normal circulation in the human body. Now, when the oxygenated blood comes from the left ventricle to the vital organs. One of the vital organ is kidney. So kidney receives the blood. If I want to precisely explain that the condition where the RAAS is activated, we need to understand that there is a situation where kidney will get less blood supply. The condition which is known as renal hypoperfusion. So the first condition where the renin releases in the blood is when the kidney is getting less blood from the systemic circulation. When the renal hypoperfusion occurs, or when the renal blood flow decreases, or when the renal pressure decreases. So in the condition where the renal blood flow decreases, there is a certain mechanism in the kidney. In order to understand precisely, let us understand by taking one nephron out of the kidney. So if we say, what is the function of kidney? So kidney is mainly purifying the blood, right? So kidney is doing all this function because of the nephron. One kidney is having 1.2 million nephron, and two kidney we have in a human body, so we have 2.4 million nephron. So let us understand the renal hypoperfusion induce renin secretion by simply understanding the mechanism of renin release using one nephron. So I have taken one nephron out of kidney, and I'll explain that how renin secretion occurs. Now this is a nephron. This is afferent arterioles. Afferent arterioles will take the blood from the major systemic circulation to the kidney, and this is efferent arterioles. These efferent arterioles will pull the blood from the nephron. So when the afferent arterioles is forwarding the blood to the nephron and the efferent arterioles is taking the blood from the nephron, if there will be decrease in the blood pressure, or if there is a decrease in the blood supply, there is a pressure sensing device in every nephron. So every nephron is basically having a pressure sensing device. This pressure sensing device will sense the fall in pressure in the nephron, and it will stimulate certain cells present in the nephron to release the renin. So to understand that, these are the afferent arterioles, and these are the cells which is present in the afferent arterioles, which will sense the fall in blood pressure. Basically, these cells are known as blood pressure sensing device. So this blood pressure sensing device are basically sensing that some of the fall in blood supply to the kidney has occurred due to some reason. And due to that condition, this particular cells start secreting renin in the blood. So from this juncture, from this specialized cell which is sensitizing the fall in blood pressure is releasing renin in the circulation, and this renin will go to the systemic circulation and stimulate the renin-angiotensin-aldosterone system. Now this specialized cell which is sensitizing the fall in pressure and releasing renin are known as baroreceptor, and this baroreceptor are having pressure sensitizing system. The second name of this cell is polkissen cells. Polkissen cells. These cells are known as polkissen cells. These are basically vascular smooth muscle. These are the smooth muscles which is present in the afferent arterioles. They are known as polkissen cells. They have a baroreceptor mechanism, so they sense the change in pressure and then they release the renin. The second mechanism why the renin is released in the blood is through the distal medulla. So if the blood comes to the nephron through afferent arterioles, it gets filtered over here. Blood will get filtered in this area. When the blood is filtered, sodium will be absorbed from the blood. If the sodium is excessively present, it will be eliminated. Now sodium, when it passes through this area, here there are certain kind of cells which will be reabsorbing the sodium. 70% of the sodium will be reabsorbed in this area, and rest of the sodium will be reabsorbed in this area. This is ascending loop of Henle, where the 25%, further 25% sodium will be reabsorbed. Rest, if you sum up this 70 plus 25, around 90 to 95% sodium will be reabsorbed. But the 10%, 5 to 10% sodium which is remaining will travel to this area. Now in this area, when the sodium is going to the distal starting part of the distal convoluted tubule, this is the starting part of distal convoluted tubule. This area, there is certain dense cells are present. These dense cells which is present in this area are nothing but sodium sensiting cell. So these cells are sensitizing when the sodium concentration get fluctuated. So when 10% sodium comes over here, these cells will sensitize the concentration of sodium and they found it normal. But when the blood supply to the kidney get decreased, there is a less blood coming to this area. When the less blood is coming to this area, the blood which is filtered having less sodium during this particular juncture, when the sodium is passes through, the concentration of cell which is reabsorbing the sodium is same, but the sodium concentration which is coming through the filtration is decreased, and thus major sodium will be reabsorbed over here, and by the time the fluid reaches to the distal convoluted tubule, there will be further fall in sodium concentration, and that fall in sodium concentration is sensitized by this dense cell which is present in the initial part of distal convoluted tubule. This dense cell, because they are densely present, you can see over here, this is known as macula densa. These cells are known as macula densa. This macula densa will sensitize the fall in sodium concentration, and again this will also release the renin in the blood. So this is another cell which is releasing the renin in the blood. Now these two cells are connected together via connective tissue cells. This polkissen cells and macula densa cells are connected via connecting tissue cells, and they both combines together with the connective tissue cells and form one apparatus called Juxta glomerulus apparatus. This apparatus is known as Juxta glomerulus apparatus, which is combining of polkissen cells near the afferent arterioles and dense cells in the initial part of distal convoluted tubule. They both are connected with the connective tissue cells and combinedly make the Juxta glomerulus apparatus, which release renin in the blood. Because this whole system is situated in the kidney, it is intra-renal local mechanism of renin secretion. Now apart from this, when we discuss about the systemic release of renin, as we have seen the circulation over here, baroreceptor is present in the polkissen cells area, where we combinedly say this area is Juxta glomerulus apparatus. One baroreceptor is present over here. Baroreceptor is a blood pressure measuring device which is present in each and every nephron. Second baroreceptor is present in the carotid sinus. This is carotid sinus. In this carotid sinus, if there is a fall in blood flow coming out of the heart, carotid sinus will sensitize that fall in blood pressure, and this will give signal directly from this area to the vasomotor center present in the brain. And when the vasomotor center which is present in the brain gets stimulated, it releases norepinephrine through preganglionic neurons. So vasomotor center, when stimulated, it start secreting sympathetic outflow, and sympathetic outflow when start secreting, it majorly secretes norepinephrine. This norepinephrine which is secreted will directly go to this again Juxta glomerulus apparatus, where the beta 1 receptor is present, and we all know that norepinephrine is the hormone for the sympathetic receptor, which is beta 1 receptor. So here in this Juxta glomerulus apparatus, beta 1 receptor is present. This norepinephrine will go and bind to the beta 1 receptor and directly stimulate the renin through this systemic mechanism, which is sensitized. The fall in blood pressure is sensitized by carotid sinus, It stimulates vasomotor center in the brain. Vasomotor center in the brain starts secreting norepinephrine through the pre and post ganglionic neuron. This norepinephrine goes and binds to the beta 1 receptor present in the nephron, and the nephron starts secreting renin. So three different mechanisms via renin comes in the blood. One mechanism is via polkissen cells, which is baroreceptor. Second mechanism is via macula densa signal. Third mechanism is via vasomotor center stimulation through carotid sinus. Fourth mechanism is there is a fall in serum electrolyte concentration. If there will be imbalance in sodium concentration, calcium concentration or potassium concentration, there will be again fall of sodium, fall of potassium and fall of calcium in or out of the cell. It will be sensitized again by the nephrons present in the kidney, and that abnormal electrolyte concentration in the human body will also be responsible for the release of renin. So this is how renin will come to the systemic circulation. These four mechanisms are responsible for renin release in the circulation. Now, as we know, renin is an enzyme. So this renin enzyme needs a substrate. So now we need to understand that what is the substrate for renin. So let us understand the concept where we find out that what substrate are present in the human body where the renin will go and bind. So this is a liver. Now we all know that liver produce the protein. Liver produce globulins. Globulin is one of the major protein in the human body. Except gamma globulin, which is produced by the plasma cell, all other globulins are produced in the liver. So the liver is producing one of the major component in protein, that is angiotensinogen. This angiotensinogen is synthesized by the liver and secreted in the blood, and in a normal human also we have angiotensinogen circulating in our blood. Continuously it is circulating. But when the renin comes from the juxtaglomerular cell, it comes and binds with the angiotensinogen, and it converts angiotensin to angiotensin 1 in the presence of renin. Now this angiotensin 1, again now angiotensin 1 is circulating in the blood. When the angiotensin 1 circulating in the blood and passes through the lungs is having one enzyme called angiotensin converting enzyme. Now if you want to learn that where exactly in lungs angiotensin converting enzyme is present, it is basically present in the pulmonary capillaries endothelium. Pulmonary capillaries are the capillaries present in the pulmonary system. Alveoli is a different component of the lungs. Alveoli doesn't have blood, it is having air. Pulmonary capillary is having blood, and because the blood is passing through the lung, this pulmonary capillary is having endothelium, and this endothelium is having angiotensin converting enzyme. So whenever blood passes through these capillaries, angiotensin converting enzyme will convert this angiotensin 1 into angiotensin 2 in the presence of angiotensin converting enzyme. Not only conversion from angiotensin 1 to angiotensin 2 is done by angiotensin converting enzyme, but along with that, it will also deactivate metabolism. It also deactivate bradykinin. Bradykinin is a vasodilator present in the human body. This angiotensin converting enzyme converts angiotensin 1 to angiotensin 2 and inactivate bradykinin. So because it is converting angiotensin 1 to angiotensin 2, which is vasoconstrictor, it is promoting the blood pressure or increasing the blood pressure, and bradykinin is vasodilator. Inactivating bradykinin, angiotensin converting enzyme is also enhancing the blood pressure. So dual mechanism this SE is doing, which is causing rise in blood pressure. Now when the angiotensin 2 is formed through this process, what are the actions of this angiotensin 2? We need to understand these actions in order to understand how blood pressure is increasing. So in the next session, we will be discussing about the different physiological action of angiotensin 2 in the human circulation. As we had discussed, the renin, which is released from the Juxta glomerular apparatus, goes to the blood circulation where it finds angiotensinogen, which is secreted from the liver, converts angiotensinogen to angiotensin 1. This angiotensin 1, when passes through the lungs, it will be converted from angiotensin 1 to angiotensin 2 in the presence of angiotensin converting enzyme, which is present in the pulmonary capillaries endothelium. And this angiotensin 2 will be circulating in the human blood. Along with that, this ACE, angiotensin converting enzyme, also inactivate bradykinin, which is potential vasodilator. Now, this circulating angiotensin 2 will have some systemic action. So, we will understand what angiotensin 2 will do in the human circulation. Now, this angiotensin 2 is having high affinity or receptor in the smooth muscle, and our blood vessel, whether it is venules or arterioles, it is having a smooth muscle. So this angiotensin 2 will goes to the venules and it leads to venoconstriction. Angiotensin 2, when binds with the receptor present on the venules, it leads to venous constriction. Now again, if you remember the factor affecting blood pressure, which we had discussed in the previous video, with the link is given here, you can understand that when the venous system is constricted, when there is a venous constriction, there will be increase in the venous return. When the venous system is constricted, there will be increase in the venous return. If the venous return increases, it increases preload. If it increases preload, it increases stroke volume. If it increases stroke volume, it increases cardiac output. If it increases cardiac output, it increases systolic blood pressure, which we had precisely discussed in the previous video. So, this is how angiotensin 2, when binds to the venules and do venoconstriction, it leads to increase in the blood pressure. Similarly, this angiotensin 2, if binds to the arterioles, when it binds to the receptor present on the arterioles, it leads to arterioconstriction. This arterioconstriction, if you have understand the concept of preload and afterload, then you can understand arterioconstriction is increasing the resistance against the blood which is coming from the major systemic artery, and thus it is increasing afterload. So if afterload is increased, if the afterload is increased, it lead to increase in total peripheral resistance, and thus it lead to increase in diastolic blood pressure. So this angiotensin 2 via venous constriction and arterioconstriction, it leads to increase in systolic as well as diastolic blood pressure. This angiotensin 2, when it goes and bind to the adrenal gland, so adrenal gland is also having a receptor for angiotensin 2, and this angiotensin 2, when comes and bind to the adrenal gland, specifically in adrenal gland, it binds to the adrenal cortex region. Adrenal gland is having adrenal medulla and adrenal cortex. Adrenal cortex is responsible for secretion of aldosterone, and adrenal medulla is responsible for secretion of catecholamines. So this adrenal gland is having adrenal cortex, which is again divided into 3 layers. One of the layer is zona glomerulosa. This zona glomerulosa is responsible for secretion of aldosterone. So this angiotensin 2 via adrenal cortex and zona glomerulosa, it releases aldosterone in the systemic circulation. Now what aldosterone will do, let us understand in detail. This aldosterone which is secreted through the angiotensin 2 mediated consequences from the adrenal cortex will go and act on nephron. Now what it will do, this aldosterone will increase sodium, potassium ATPase concentration in the nephron. Now, due to increase in sodium potassium ATPase concentration in the nephron, extracellular cells which is present in the nephron starts throwing sodium from the cell to the interstitium. Start throwing sodium from the cells to the interstitium, and the extracellular cell which is present in the nephron are deprived of sodium. So when the filtered blood passes in the nephron, which is in the form of urine, when it is passing through the nephron, concentration of sodium will be higher in that region and concentration of the sodium is lower in the extracellular region. Why? Because in that extracellular region, let me explain if you don't understand with this figure. This is the wall, this is the nephron, and this is the extracellular component of the nephron. Now this aldosterone, through gene activation, increases the sodium potassium ATPase in this region. Due to that, sodium which is present inside the cell will be thrown out via the sodium potassium ATPase transporters, and thus sodium concentration goes down and potassium in extent to sodium increases. Now when the sodium containing fluid passes from this region, this sodium concentration will be higher over here and sodium concentration will be lower over here. So as a concentration gradient concept, higher concentration to lower concentration solute moves. So the sodium will be coming back from the filtered urine, it will be coming back to the cell, and from the cell it will be coming back to the interstitium, that is nothing but blood. Thus this aldosterone retains sodium in the blood. Now, as a thumb rule, sodium will also try to retain the water. So wherever sodium goes, water follows. It is said that wherever sodium goes, water follows. So when the sodium is retained via this mechanism, water will also retain in the human body, and sodium water retention is the action of aldosterone. This sodium water retention by aldosterone leads to increase in blood volume. Again, you remember the chart. If the blood volume increases, it increases venous return. If the venous return increases, it increases preload. If the preload increases, it increases stroke volume. If the stroke volume increases, it increases cardiac output. And if the cardiac output increases, it increases systolic blood pressure. So this is how angiotensin 2 mediated aldosterone release leads to increase in blood pressure. Let us understand the angiotensin 2 action in the different components, namely central nervous system, peripheral nervous system, heart, kidney and intestine. So let's start with the central nervous system. So this angiotensin 2 is having a receptor in the hypothalamus, and through hypothalamus it stimulates the posterior pituitary gland. So when the angiotensin 2 goes to the central nervous system via hypothalamus and posterior pituitary gland, it secretes antidiuretic hormone. Now this antidiuretic hormone is having action in again nephron. It increases the permeability of the nephron in the collecting ducts, and because of the increase in the permeability, there will be retention of water, and due to the retention of water, antidiuretic hormone keeps on retaining water, and because of water retention, it increases blood volume, and because of increase in blood volume, as we discussed earlier, it increases systolic blood pressure and to some extent diastolic blood pressure. So angiotensin 2 via central nervous system secretes antidiuretic hormone, and this antidiuretic hormone increases the permeability in the collecting ducts, and thus because of increase in the permeability, water will be retained more, because when the permeability is increased outside the duct, there will be high concentration of solute, and because of the high concentration, there is a high osmolarity. Inside the duct, there is a low osmolarity, and to maintain the osmolarity, low osmolarity, the water from the low osmolarity will move towards the high osmolarity to maintain the isotonicity. And this is what the mechanism of antidiuretic hormone, and due to this mechanism, there will be rise in blood volume. If there is a rise in blood volume, rise in venous return, rise in preload, rise in stroke volume, rise in cardiac output, and rise in systolic as well as diastolic blood pressure. The second important action of angiotensin 2 is on peripheral nervous system. So when the angiotensin 2 binds to the autonomic nervous system, or the action of angiotensin 2 in the autonomic nervous system is stimulation of pre as well as post-ganglionic neuron. Now say for example, if I zoom this post-ganglionic neuron will release norepinephrine in the blood. This norepinephrine, how this norepinephrine act further? So now this norepinephrine works via two mechanisms. This norepinephrine will be bind to the norepinephrine receptor which is present in the cell, and rest of the norepinephrine will be reuptake back to the vesicle. See, norepinephrine which is released from the postganglionic neuron vesicle, this norepinephrine releases, certain norepinephrine binds to the norepinephrine receptor which is called adrenergic receptor which is present in the human blood, and the rest of the norepinephrine will be retained back, or it will be reuptake. It is called reuptake of norepinephrine. Now, when the angiotensin 2 is released, it will keep on increasing norepinephrine. So the concentration of norepinephrine rises. Apart from that, it will stop reuptake of norepinephrine in the vesicle. And this is why release is also increased, reuptake is also decreased. There will be very high concentration of norepinephrine in the systemic circulation. And wherever there is a receptor for norepinephrine, which is in the heart, in the nephron, in the smooth muscle, wherever there is a receptor of norepinephrine, this norepinephrine go and binds. And when it binds to the norepinephrine receptor, it leads to, say for example, if it binds to the vascular smooth muscle, it increases the preload and afterload via venous and artery constriction. If it binds to the heart, it goes and binds to the heart, it increases heart rate, it increases contractility. If it goes and binds to the kidney, it releases renin again in the systemic circulation, and the RAAS system will be activated. It also stimulates the aldosterone release. So this is how this peripheral nervous system secretes norepinephrine, and norepinephrine mediated blood pressure will rise. Next is angiotensin 2 has a direct action in the heart. Angiotensin 2 in the heart will increase contractility. It increases contractility. So when the contractility in the heart increases, it increases stroke volume. And if it increases stroke volume, as we had discussed earlier also, it increases cardiac output and it increases blood pressure. And the last is angiotensin 2 has a direct action in the intestine and kidney, where it retains the water. Similarly, like ADH and aldosterone, direct action of ADH in the kidney and intestine is retaining the water, and retaining the water leads to increase in the blood volume. Increase in the blood volume leads to increase in the blood pressure. Now the important point I want to explain over here is this angiotensin 2, if keep on increasing in the systemic circulation, it has a negative consequences in the heart, because this angiotensin 2, when it goes to the heart along with the aldosterone, it changes the morphology of myocytes. It changes the morphology of myocyte up to some extent that myocytes are now hyper grown. There is a secretion of certain growth factor along with the change in morphology in presence of angiotensin 2 and aldosterone, and this leads to cardiac remodeling, and this cardiac remodeling leads to congestive heart failure. And that is why angiotensin 2 blocking drug is majorly prescribed in those patients who are having a chance to develop heart failure. Because if angiotensin 2 is keep on increasing in the human body, it is definitely going to harm the heart, contractility as well as remodeling of heart, and the myocyte structure will change. Then myocytes will no longer helping heart to pump the blood, and that is why angiotensin 2 blocker or angiotensin converting enzyme is highly prescribing in those patients who are having high concentration of angiotensin 2 and aldosterone. So this is what angiotensin 2 will do in the human body. These are the different angiotensin 2 physiological actions.